Today's speaker is going to really keep us at the edge, and we will all be listening to her. I'm very happy to introduce Professor Usha Vijayaraghavan, a well-known personality. In fact, needs no introduction, but still, the organizers asked me to introduce her. Professor Usha Vijayaraghavan is the Dean of the Faculty of Biological Sciences at the Indian Institute of Sciences, Bangalore. A distinguished academic researcher, she has made significant contributions to the fields of molecular biology and cell biology with a focus. Sorry for that. Understanding the molecular mechanisms that govern cell growth, differentiation, and development. Her research group at IIC investigates the regulation of complex gene networks, particularly in the context of flowering and plant architecture in rice. Her work uses genetic and genomic tools to understand the mechanisms that control plant growth and development. She has guided many PhD students and has been involved in various national and international collaborations aimed at advancing biological research and education. Professor Vijayaraghavan is a fellow of the Indian National Science Academy, the Indian Academy of Sciences, and a JC Bose Fellow of the Department of Science and Technology. She has received numerous awards, including her Swaroop Medal, Indian National Science Academy, the National Bioscience Award offered by Department of Biotechnology, and the C.V. Raman Award, Karnataka State Science and Technology Council, for her outstanding contribution to plant biology. Madam, they have also given me another challenge to really fix the time within the time limit. Your talk is originally scheduled for 40 minutes. If you can reduce it a little bit, and we can save time so that we can go to the next.
So this is a wonderful opportunity, and I'm thankful to both Arjit and Soumya for putting this exciting workshop together that covers very many diverse aspects of how soils, microbes, and microbial and communities, which are macrobial communities in the soil, influence plant health and plant productivity. So in today's talk, I'm going to take you to some very fundamental biology of how it is that plants actually develop in the first place, right? And anything that I say will in some way impinge on many of the crop plants, many of the vegetable plants, and many of the non-crop plants, which are the bulk of the plant kingdom that make our ecosystem. So I'll give you some glimpses on my lab of what my lab is doing to trying to understand how the development of the rice plant occurs, particularly how is it that they make flowers. Okay, so as all of you know that flowers and fruits, flowers are the progenitor for all grain, seed, fruit, anything that we consume. Flowers are often the place where reproduction occurs, and therefore they're important for the plant in its life cycle and the evolution of plants itself, right? So from a very anthropomorphic view, we'll say, oh, their flowers look so beautiful, so we will breed them for their beauty. We'll breed them for the kind of colors they have, the kind of diversity of organ forms they have, or we will breed them because they make a seed or a fruit or a grain, which is of a nutritional value for us. So flowering plants, where did they come from? How are they there in every single ecological niche? This is a big question that Darwin pondered about. Okay, you all know Darwin? I mean, you can't be a biologist without knowing a giant like Darwin, right? So what did he call this? He called it the abominable mystery of flowering plants. Where did flowering plants come from? So it turns out that flowering plants are actually rather recent entrants to life on Earth. How long have we had living beings on Earth? Eukaryotic living beings on Earth. Anybody wants to take a guess? 5 billion years? 10 billion years? 2 billion years? Half a billion year? Eukaryotic? Huh? Nikhil's muttering something. Okay. Anyway, so eukaryotic beings are those where multiple cells come together to give the organism's form and function and control its reproduction, etc. Prokaryotes and archaea existed prior to eukaryotes, but all of this is at least 1.7 billion years. So in 1.7 billion years, 200 million years is nothing. It's a very, very short time span. But yet, this particular group of plants, which are the angiosperms, occupy every niche, like I mentioned, and they have a huge diversity in form, and particularly the diversity which typifies the angiosperm is the flower. Where is the flower made? How is it made? And what type of organs does it make? There's a huge amount of diversity there. So it's a fantastic question on trying to understand fundamental mechanisms which must be shared in the ancestral evolution, during the ancestral evolution of the flowering plant. Because all angiosperms share flowers, and in flowers only the reproduction takes place. So there is a very, very ancient ground plan of making a flower which was there established in the ancestral angiosperm. Somehow that ground reality of making a beautiful flower is tweaked here, there and the other to give you the diversity in the form and function, right? So that's what we'll try to understand using a model. And for every biologist, every geneticist, you usually will learn a lot by just observation, right? You need to observe the process and see what is going on. Then you can frame the question relative to the process to see how can you perturb the process? And does the perturbation inform you of how the normal process occurs? That's how geneticists will view it. So now the question is, probably all of us ate the mushroom rice or the plain rice or whatever, you ate the rice. But where the hell did it come from? That's the question. So if you look at a rice plant, just like any other seedling, when you germinate it, the seedling already has in it a preformed small embryo, and that embryo has a group of what are called as multipotent stem cells present over here called the shoot meristem. That's all that's there. And it will have a couple of embryonic leaves. In the case of rice, it has about 4 or 5. Monocot cereals have 4 or 5 embryonic leaves. Everything else about the plant is post-embryonic development. And what is that post-embryonic development? It will give rise to more leaves from the side, and in the axil of the leaf you will have more meristems. What are meristems? They're groups of cells which have no differentiation program of their own, but they have the capacity to develop into different structures. But on the perception of the right time, that is the age, the photoperiod, the conditions that it's present in, its nutritional status of course comes in, we understand less about that, and the hormonal status of the plant, the rice shoot apex, which is making leaves and axillary meristem, now switches into something which is a flowering stem. A flower bearing stem is called an inflorescence. So that inflorescence could be an inflorescence which is going to give you the date palm, or could be an inflorescence which is going to give you a cauliflower, which is also an inflorescence, or can also give you a broccoli or whatever. But in the case of rice, it's called the panicle. It generates now rapidly growing cells which make multiple orders of new meristems. So these are called branch meristems. These branch meristems then give secondary branch meristems, and ultimately they will make a single flower. The single flower is made on the final short branch. The flower is entirely closed. So it is not pretty. When you look at it, you walk past it before it has come out, all of this development is occurring within the enveloped leaf sheath. So you won't even know that the rice plant is flowering. But once this differentiation and flowers are made, then the stem elongates and comes out of the plant, and that's when you will see driving past Tanjavur fields, some yellowing in the entire field will look a little bit yellow. That's because you will be able to see the pollen in the grain, pollen in the floret. So the floret is mostly closed, unlike the open florets of hibiscus or rose or lily, etc., where the flower is open. The flower opens out, and you can see the organs of the flower, you can see the male gamete-bearing stamens, you can see the carpel, etc. Here it's a completely closed structure. This closed structure will open for a regulated amount of time, at the right time and right place, remember we were talking about time and place, enough to open to allow the stamen to elongate and the shedding of the pollen to take place. After the pollen is shedded, it will collapse and the flower will close again. Both the opening and the closing are developmentally controlled events. So there's a lot of very nice biology that one can ask in saying that, okay, it is a flower, but these many organs which are present in the rice flower look very different than that of a mustard or a tomato or a hibiscus and so on. What are these organs and why are they different? So the two outermost organs which are performing what might be the role of a sepal, that is to cover and protect the inner organs, look very different. They're very thick, leathery, have lot of epicuticular waxes and extensions. And secondly, they are closed because they are interlocked structures. So it's a mechanically hooked structure which is tightly closed. That opening is facilitated by two small structures called lodicules, which are thick and fleshy structures, and they are asymmetrically positioned. They're positioned in only one part of the flower. So imagine you have, I will show you more of that, on only one side a fleshy structure. If that swells, it can push open and serve as a lever. That allows for the stamen elongation to take place. So the structure of the organs which are the non-reproductive organs, the stamen and the carpel, is very different than that what you will see in the dicot species. But this structure of the rice flower which I have described is actually common to all grasses, all cereals. So what I have described to you also applies to maize, also applies to barley, also applies to foxtail millet, setaria, anything. All grass florets, finally, when they are made, they will be this structure. How many florets they will make, how branched this inflorescence will be, all that is variable even within the grasses. Right? So now, how are floret organs made? How do you address this question? So I said 180 million years ago, flowering plants came. So to address the fundamental question of why is it that all floral organs are positioned in this stereotypic pattern, there were 4 or 5 people who took on this big challenge. Nothing was known about it. Why is it that in all flowering plants, sepals are on the outside, petals are inside to the sepals, inside to that are stamens, and inside to that are the carpel? This is the invariant pattern. There's an invariant pattern in 350,000 families of flowering plants which are extant, that is present today. Forget about the extinct. So why is this design fixed? There must be some ground rules which are common to all flowering plants. To address what are those ground rules, Enrico Cohen, Elliot Meyerowitz, Heinz Sadler, and Martin Corneve used 2 models. One was Arabidopsis and the other was Petunia, and tomato, 3 models, to ask, can we make mutants, genetic mutants, where this pattern is altered? Usually genetic mutants tend to be loss of function. That is, you lose a particular gene function, and if that gene function is important for this pattern, then the pattern will change. And then you trace back and see what is that gene function which was lost in the mutant, and then infer what it does in the wild type. Through about 30 plus years of their work, their iconic model, which was called the ABC model, is now very well established, that there are master regulatory transcription factors which act in a combinatorial mode in order to give identity to specific regions of the floral meristem where organs are going to develop. So if you do not have these regulatory switches, the right downstream events of development are not triggered and something else happens. So the combination of, these are color coded over here in different colors. The original model had only 3 factors, but said that this is easily expandable, and that's true. It's expanded to about 5 different classes of factors now. And using either Arabidopsis or tomato or Petunia as a model, the same rules apply. Class A factors, when they are present along with their mediator complex, will give rise to sepals. Class A plus class B factors together will give you petals. Class B plus class C factors will give you stamens. Class C factor will give you carpel, and where class C and class E are present together, D are present together, you will get an ovule. The model also showed generically that class A and class C are antagonistic to each other, and therefore wherever A activity is present, C will not be present, where C is present, A will not be present. So by a simple combination of 5 classes of master regulatory transcription factors, you can set up the zip code. A zip code of how in the young floral meristem you will make a sepal, petal, stamen, carpel and an ovule, right? So this model, the reason it has to be very, very conserved is because Arabidopsis is not at all, is not even in the same family as Petunia, which is not in the same family as tomato, but yet the mutations gave the same phenotype, and the genes, once they were cloned, were all homologous to each other. So this said that these are highly conserved, evolutionary conserved transcription factors. Many of these transcription factors were recognized to belong to the family defined only because of the work done in plants as the MADS domain containing factor, where the MADS domain is the one which binds DNA on their target genes and activates gene expression. MADS domain transcription factors are not present only in plants, they're present in animals, they're present in yeast. It is just that in the plant kingdom, evolution has used MADS domain transcription factors very often to control form and function. Just like in the animal system, Hox genes and homeodomain genes are used very, very frequently to drive form and function and patterning, right? Now, if all this is known in all those models which I talked about to you in eudicots, is it there in monocots, and more specifically, is it there in the cereal or the grass family? That's the question. Now, the reason this is important to ask is cereals and monocots diverged away from the dicots also about 11 to 20 million years ago. So that's a long time. In that time, what has happened? Are these genes present? Yes, these genes are present, roughly color coded, but there are many small variations which you can already see. Certain members are represented by more numbers in the case of rice. Certain others, the pattern of their expression is different. Just by looking at the yellow and the yellow here, you can tell that. And certain others, they are present in the same domain, but they've got some duplications and some variations of function. And certain members are completely new. They are contributing to carpel development in rice, but they have no carpel development function in Arabidopsis. So evolution has used the same modulators by either expanding or contracting the gene family members, tweaking their functions a little bit and recruiting new members. And it is the combined activity of this that gives you all these floral organs. So now let's go to some reasons as to why one should go deeper into the cereal model. Right? So in the first part of the talk, I'll tell you a little bit about this family, and then later on I'll tell you a little bit about this family. And I'm ready to wind up at any time given. So that yellow bar, which was labeled in Arabidopsis as CEP1, CEP2, CEP3, CEP4, are all MADS domain transcription factors which act as mediators in the complex with either A or B or C or D. They have to be present because in order to make this multimeric complex, these factors are essential. But they are entirely equivalent to each other. So when I teach, I say that that is like the vanilla ice cream. Okay, good, essential, but the toppings make all the difference, right? So any one of these 4 vanilla factors would do the classy job in Arabidopsis. So in order to see what is their effect, you need to make a quadruple mutant. If you make a quadruple mutant that was done in Marty Janowski's lab, CEP1, CEP2, CEP3, CEP4, then all the floral organ characters are gone and the flowers look like leaves. The flowers are coming where they should come, but all the organs are looking leaf-like. So you just don't know how to make the organs of the flower by the loss of one family of transcription factors. But when you now start looking at the phylogenetic tree of the sepallatas in grasses, mapped over here are rice and maize, MZ is maize, OS is rice, you will see that related to the Arabidopsis cep4, there has been an expansion. There are many members in the case of grasses. That family has expanded hugely in grasses, and there are no dicot members, in fact, no lily members even, not even monocots. So this lineage has expanded in grasses. Cep4 is represented by 3 genes in rice, but very importantly, these 3 genes have individual functions. They are not redundant with each other. Loss of even one of them derails the development of the floral organs. So loss of function mutation in OSMADS1, this wild type flower is now converted into something which doesn't look like a flower at all. It's sterile, it doesn't have stamens, doesn't have carpels, doesn't have laudicules, doesn't have lemma, palea. Everything looks like a series of gloom-like structures. So that means loss of just that one gene is enough to derail the program, whereas its closest relative CEP4, loss of CEP4 alone will not give you the phenotype. You have to lose CEP1, CEP2, CEP3 also. So this was a gold mine to realize that in that 11 million years to 18 million years time, this gene has become very much more important in rice than it was in the case of Arabidopsis. Right? Now to illustrate the point, its closest cousin is MADS34, and the MADS34 mutant has a completely different phenotype. It doesn't have the same phenotype. So two cousins, which are duplicated genes, have evolved to occupy unique functions. Right? So then we also need to know, all right, if it is so important for making a flower, where is it expressed? And its expression is from the very earliest stage that a flower primordium is being made. So this dome of cells has no differentiation. It's just a group of meristematic cells. But MADS1 comes on over here, but not in the inflorescence meristem and not in the vegetative meristem. So time and place, again very important. So yes, MADS1 is an important regulator, but it itself is regulated by many factors upstream to it. So it is turned on over here, and as the flower is developing, its own expression gets controlled. It's expressed in the 2 outer organs, and in the very tip where the stem cells are present. So it is under control of other things, it is controlling other things, and it is continually being controlled itself, right? And it is the correct sequencing of all of these that gives you the right flower. So we started to ask, how does it do so? If in the absence of OSMADS1, all floral organs will become BRAC-like, what are its target genes? Because it is a transcription factor with a MADS domain. So what did we do? We took young panicles which are in the 2 millimeter to 20 millimeter. So these are deeply embedded recessed tissues. So you have to dissect and collect these meristems for months to get enough tissue to do chromatin immunoprecipitation using antibodies that you have to raise yourself, because you cannot order them from any catalog which is there in the market. And using these antibodies, we asked what are all the genes to which this transcription factor is bound by doing chromatin immunoprecipitation and mapping to what locus it is been found to. Being a transcription factor, it's present upstream of genes, upstream of the transcription start site. It's present at the transcription start site, but very interestingly, it's also binding sites are within the gene, right? So about 30 to 50% of its binding sites are genic. So that tells that many cis regulatory elements which control the expression of the gene are not necessarily only upstream. These elements are also important. So yeah, I'll finish with this one. I won't go to the second. Okay. So now having the mutant, having the binding sites, we could ask one more question. Okay, the transcription factor is bound at the locus. Maybe it's not doing anything, it's just sitting there. Okay, so some of you may not be listening, you're not compelled to listen. You can be present, you can mark your attendance, but you may not be interested, that's all right. But some of you might be interested now, and some of you may be interested maybe after 1 hour when you're thinking about it and you will get activated, right? So presence is not assurance that there is action, right? So to determine that, what we did was determine deep transcriptome analysis, but did this in developmental windows. We took panicles which were just 2 to 5 millimeter when the flowers are just forming, that is a dome which I showed you at that stage. We took those which are at 5 to 10 millimeter when organ primordia are just going to come out, and then 10 to 20 millimeter where organ primordia are just elongating. Remember, even at this 20 millimeter stage, still there is no pollen, anther is just about coming up. So we are asking what are the early developmental programs that it kickstarts. And we did a differential gene expression between the wild type and the mutant, queried all the differentially expressed genes, and then mapped it back to those which were bound in order to find out what are the direct targets and indirect targets controlled by MADS1. So before we went into the how it is dynamically controlling gene expression, we asked in wild type plants when they are going through the process of development from early, middle and late, what is the profile of gene expression that is changing place. So you can detect clusters of gene show a certain pattern. This cluster represents a set of about 4,000 genes in the entire genome, which are high in very young flowers, go down in the middle and go down even further later. You have the exact opposite of about 3,000 genes, which are very low here, but they keep going up. So these are developmentally co-regulated genes through the process of development in the wild type flower. Now we superimpose on that what went wrong in our mutant. And when you look at cluster 1, we see cluster 1 in this early gene set, there is a huge number, about 1200 of them are activated by MADS1. So what is MADS1 doing? In these early flowers, it is activating the program of floral meristem identity, right? But it's not doing only that. I mean, one can assume that this is all that it's doing. But it's not the case. If you look now in the middle stage when organ differentiation is going on, there also it's activating, but it's activating another set of genes in this stage, and it is about 800 or so. So what are the genes that it's activating in this window? These all tend to be meristematic genes. That is, they are all homeodomain containing transcription factors which control cell proliferation. And they are very important for the meristem to maintain itself. You start with 100 cells, you certainly can't make all the organs with 100 cells. So you need to grow to 200, 300, 400, 500, but just the right number. So that all of them will get partitioned into differentiation programs. So a lot of meristematic genes are active in early stages, including some of its own MADS domain type of transcription factors. So then we ask, okay, it's controlling meristematic genes. Is it similar to what Arabidopsis flower meristem is doing? There's a huge data set available in Arabidopsis. Arabidopsis also SEP4 targets have been mapped. So unlike Arabidopsis, we found that in many cases, the activation is indirect. It activates through another activator, okay? The homeodomain factors, the number that it regulates in rice is much, much more than in Arabidopsis. It influences about 27 different genes in total, including 2 very key genes which are Knox family genes of the homeodomain transcription factors. Now here is a huge meta-analysis of experimentally proven data sets. These are not bioinformatics. These are from the actual data that we have. In early flowers, MADS1 directly activates OSH1 and OSH15, which autoregulate each other as shown by Suda et al. So Suda et al, a Japanese group, was working on OSH1 and OSH15. So they had already shown that they autoregulate each other, but what we are showing in floral meristems, there is a feed-forward network where MADS1 activates them, OSH15 activates, and this makes a feed-forward network which keeps the floral meristem on. And when you don't have MADS1, then this network becomes very weak and the meristem is collapsing, right? So these are not the only ones. They in turn can have a huge domino effect on 51 other transcription factors and chromatin regulators. So one has got an amplifying effect because it can control 51 other factors. These 3 are controlling, 52 other factors, such a way that it can give a dominating ripple effect. And between these transcription factors also, there is a lot of networking which is going on. They are controlling each other. So MADS1 activates all these transcription factors, MADS1 activates these, this one represses this, and it is this very, very fine tuning of the network that controls the property of making a floral meristem. Now, if you understand all that, does it actually tell you anything about how the rice floral organs are made? Yes, this is a goldmine of information because we can now see what are all the genes which map into specific organ primordia by meta-analysis of other people. So somebody else might have studied, for example, ARF8. They haven't studied, but I'm giving you that as an example. And they might have reported that in ARF8, stamen development is disrupted. But they don't know how ARF8 is regulated. We are able to say that MADS1 activates ARF8 and ARF8 can control the downstream process. So another such important gene is EMS1, which has got to do with male sterility. So MADS1 is directly activating EMS1 so that when the anther is made, the pollen will be fertile. So you understand many important downstream targets of MADS1, and allelic variation in any of these can contribute to when flowers are made, how flowers are made, and how floral development occurs. So I'll stop right here. I won't give the second story, which is much the same. So what are we inspired by? We're inspired by both experimentally derived data sets, which the biologist knows and can very, very accurately design tools to address that. But combine that with the expertise of computational biologists and mathematicians in order to understand how this network might work, because biologists can make only X amount of tools and probe the system. But by curating the very important biologically derived data sets, can you now make all the other connections which you will not be able to do experimentally? All right. So how do you go from making a flowering stem to a single flower? And this is I've shown only one snapshot. There are dynamic networks like this, which are constantly changing. It won't be possible to experimentally determine all of them. And it is by working together as a team that we might be able to address this wonderful challenge that biology has thrown to us. I've been blessed to have good collaborators, including Arjit, who's over here, and fantastic group of students through many years. Some of them are professors in their institutions and leading their own research teams, yet others are submitting their thesis and headed out. Thank you all.
Thank you very much, Professor. Now the floor is open for discussion. Few questions we can take up.
Usha, as usual, fantastic. Just I don't remember. So all these initial in Arabidopsis and tomato and this other petunia. So is it the same ortholog? Because this MAD is a very huge number of.
Same. So they are phylogenetically placed.
So it's same ortholog in
Same.
all these three.
It will be called homologs. It will not be called ortholog.
Homolog, right?
Homolog. So
But the same number. So if
no number you cannot tell, but we have to do the phylogenetic placement. For example, SEP1, SEP2, SEP3 is represented by OS MADS1, OS 34, and OS MADS5. The numbers will be different, but it's by phylogenetic placement.
In the tomato and in
within each family.
But when you see that between the monocot and dicot, also it is the same, or there would
no,
be
the variation is variation in the number.
In the number,
right? So 4 genes of Arabidopsis is represented by 5 in rice. So it's not 20, just one more. Yeah.
Please. Yeah, you go ahead.
I think he had something.
No, you go ahead.
Okay, he wants me to start off. So this is a lot of work, but still it looks like this MADS4 is like important transcription factor. It is really like a tip of the iceberg.
MADS1, these type of factors are called pioneering transcription.
Yeah, so there is so much more, there's a lot of feedback regulation. So each one of them require enough and equal attention, isn't it?
If MADS1 is not present, all the downstream things are falling apart.
That's true. But like MADS1, there must be many other master transcription factors too.
Yeah, it depends on what question you're asking. If you're asking the question of what controls floral identity, then it's MADS1.
Right.
But if you say what's controlling lateral root identity, it will be something else.
True. Yeah. But if the question is different, like stress or something, so
Yeah,
similar
no,
question.
the stress is not one factor. The stress will impinge on this.
Yeah.
So where stress also has to work on some developmental regulator, right?
Absolutely.
If you have given heat stress and you find that pollen fertility has gone down, what is it doing? It has to work on one of these genes, right? So
I have
we
more,
have,
but I will.
no, we have a set of 50, 60 genes which are important for stamen development. That stress has to go and act on them.
Yes.
How will you know which
Stress
is which?
also gives rise to flowering, people say.
Yes, through these genes.
Yeah.
Yeah, yeah.
Two more questions we can take. And
Thank
mostly
you,
from-
ma'am. It was an eye-opening. Like, first of all, vegetative to reproductive transition itself is
I didn't talk about that at
No,
all.
ma'am,
Yeah,
my
yeah,
question
yeah.
is related to that. It itself is complex, like 8 pathways has been identified.
Yes.
And it is such a complex, and coming from meristem to floral, you have shown that this is another complexity,
Correct.
too much complexity. So I worked in cauliflower and we exploited the FLC and FRI genes for tropicalization of temperate types.
Right.
So what is your objective for particularly breeding aspect? Like,
Okay, so I don't want to say that I don't admire breeders, I admire them, but my motivation doesn't come from breeding. My motivation comes from asking how does development occur. Since you worked with cauliflower, I'll tell you. Cauliflower. Now, why do you want to have these FLC and non-responsive mutants and so on? You want to see that you can synchronize the transforming of the meristem into a cauliflower which doesn't shoot. Okay, it doesn't spread out. So you see this ugly looking spread it out, nobody will buy that cauliflower. You want tightness, you want very quick transition from one development to the other development, and after that you want arrest. You don't want to eat cauliflower where petals and stamens and carpels are there. They are arrested in development, right? So you want to be able to address that from a developmental question. That's inspiring. You can also ask it from a breeder's perspective. So it depends on which end of the spectrum, both are equally important.
Thank you.
So some of these genes will be important as breeder's tools. For example, some of the genes which are downstream to MADS1 will control fertility. So variation in those 50, 60, 100 genes which are controlled by MADS1, one can now start asking, are there allelic variants there that contribute to fertility?